So Euglena is actually a genus name, a bit like Homo for Homo sapiens, and there are, that we know of, about 800 species of Euglena. It's a single-celled organism that has the ability to photosynthesize, but it's not a plant. Euglena is really interesting because it's related to a group of really important parasites that affect both humans and animals. And so by studying both of these groups, we can understand how this group of parasites has evolved those particular traits that allow it to invade a host and cause disease. The Earlham Institute is one of the biggest genome sequencing centres in the UK. Its speciality is genome sequencing, particularly of protists and things that are really difficult to sequence. So that's why we're working in Euglena. So my research at the moment is focused on actually building the genome for Euglena. It's quite a complicated organism. There's 3 or 4 genomes wrapped up in one cell because it's an example of a secondary endosymbiosis. The first endosymbiosis event was the addition of mitochondria to eukaryotic cells, and then plants got their own chloroplast, which allows them to photosynthesize. Euglena can photosynthesize, but not by eating chloroplast. It does that because it ate an algae that already had chloroplast. Because they're so difficult to study in a lab environment, we have to resort to molecular techniques to study them, things like genome sequencing, to compare and contrast between free living groups, Euglena, and parasite groups. So it's a very complicated issue, and actually generating the genome for this organism would be really useful in comparing and contrasting with related parasites and allowing us to understand how those parasites cause disease.